Okay. Hello, everyone. Welcome to the problem solving session 6 of NPTEL course, which is cell biology. Let me just bring you to the main slide. Right. So this is the problem solving session 6 for this course. We have had 5 such sessions previously, and we were discussing. So if anyone is new to this course, new to this problem solving session, what we do in this problem solving session is basically try to go over the topics that you covered in this week's notes. And what I want to do is give you some questions so that it can help you understand the topic of this week's course better. And additionally, I will also be covering, like questions from all the different topics, all the different 4 lectures from previous year's assignment, previous year's exams, so that it gives you a good grasp over the topic that you're going to be anyway studying in this week, right? Okay, so let me just begin with this week's division, if you remember. So if the 6th week mainly comprised of, sorry, the nuclear and chromatin organization. So where basically the first lecture of that week was about nuclear organization, how the organelles and how the nucleus is organized. The second lecture was about chromatin organization, so how the chromatin is organized. So you have nucleus, the organelle nucleus, and then in that you have several other components and compartments. And then in nucleus you have DNA, and which is organized at a certain level. There is some compaction, there is some protein interaction. So how is that organization taking place? And then after that, the third lecture spoke about SMC proteins and chromosome organization. So you can see from a single strand, you go to move. So chromatin with proteins are called chromosomes. So then you go to chromosome organization. And because we have already studied about DNA replication, we know about anaphase and telophase and cell cycle, we know how important the SMC proteins are, which are structural maintenance complex, which are responsible for the integrity of your DNA, which finally comes up as this packed molecule. So we will be doing about that. And then one of the important process that is carried out by this SMC protein, which is also called DNA loop extrusion, which is basically going to cover how the proteins which are which associate or interact with DNA to make up the DNA molecule, which is called histones, if you remember, how that packaging takes place is what's going to be discussed. And it was a short lecture, if you remember. So the main focus was about SMC and about the organization at different levels, right? Okay, so let's begin with the first topic, which is nuclear organization. Now, as I mentioned, nuclear organization basically refers to the spatial arrangement of everything that is found in the nucleus. So you have chromatin, you have nuclear bodies, you have regulatory bodies inside the nucleus. And what this topic basically tries to cover is how this arrangement of these different components of the nucleus influence the gene expression, because depending on your chromosomes availability, so gene expression is basically transcription of your gene. And for transcribing your gene, you need to have the access, or the DNA, the gene has to be accessible to RNA polymerase, to transcription factors. So how these are accessible to those proteins is dependent on how they are arranged. So if you remember heterochromatin and euchromatin, so that's what we are trying to refer to here, that the arrangement of the nuclear components influence the gene expression, influence DNA replication, because they were super coiled. Otherwise, in normal condition, they have to be first opened up. Then you have to have the fork formation for DNA replication. So all this requires remodeling of your DNA. Remodeling basically means in your native form right now, DNA is tightly coiled and packed. You need to open this structure up so that you can replicate this DNA and then put it into a new cell and then continue your cell cycle. So because you have to open it up and again close it again and again make it compact, that's all called remodeling, right? So we have to do that also. And then it's of course part of DNA repair as well, right? Now, so basically all of these are influenced, or all these processes take place depending on how your DNA is organized. And the organization of DNA is very structured and it is compartmentalized, as we will see. Okay, so let's talk about chromosomes first, or maybe let's talk about, as you can see here, there are so many names that are, just a second. Yeah. So all these topics or all these names, nucleosomes, heterochromatin, euchromatin, NPC, and all these different proteins, which I am sure you would have read somewhere or the other, microfilament, intermediary filament, all of these are part of the nuclear organization, and all of these have a role in how the nuclear is packed. So let's first begin by understanding the simplest way how you can explain or how you can represent nucleus, right? So the first thing that you have in nuclear is the nuclear envelope. This envelope separates the nucleus from the cytoplasm, right? Because the cell outside this is cytoplasm. So this nuclear envelope that you see here, or the nuclear membrane, it demarcates the region of nucleus. Okay. Then you have the nuclear lamina, which is basically everything inside the nuclear membrane. So anything here is nuclear lamina because it is formed of lamin proteins. Okay. Then you have let me try to show you this. So you see lamin A and C here, lamin B. These are the lamins that I was talking about, right? Now, if you remember from your lecture notes, you had some regions like uh, lamina associated domains, nuclear associated domains. So these are those regions where your chromatin region interact with lamina. Why do you need that? Because you need your DNA to be packaged and stationary in a region. So these lamin gives the region where chromatins are stationarized, and they are basically housed there. Now, what do these domains do where they are attached with the, or where they're interacting with the chromatin? They make these gene poor, and they make it heterochromatic, and they make it transcriptionally silent. And it's all very easy to understand. First thing is, if it is lamina associated, basically it is already interacting with the DNA. It is making it something called heterochromatin. Heterochromatin is basically also called gene poor. Gene poor, in other words, would mean that there is not much gene available to do transcription, and that can only happen if your genes are hidden from the nuclear or transcription machinery, right? So if it is already interacting with the histones, and if it is already interacting with these proteins of lamins, and it is already coiled, the genes are not available on the surface for the transcription factors to come and interact. That's why these regions which do not get transcribed, they're called gene poor or transcriptionally silent, right? So that's one thing. And then basically what you see here is you have this nuclear pore complex. You have nucleolus, which is the inner material. You have the nucleoplasm, which is uh, so this is your nuclear membrane. This is your nuclear plasm. You have lamin protein here. You have the chromatins here arranged. There's a nucleolus, which is the central part, and then you have this nuclear pore complex, which are very important, right? So let's talk about nuclear pore complex, which is also otherwise in mostly in textbook you will find the name as NPC. Okay, so now this nuclear pore complex, as the name suggests, it's a pore on the lining of the nuclear membrane, and it's very and has a very important role to play, right? Why is it? First of all, this pore complex, the name complex, wherever in biology, in any method, in any structure you find complex, just remember it's multimer of protein or multimer of different biomolecules together, right? So right now we are talking about nuclear pore complex. It basically means that the pores so the pore is lined by multiple proteins together, right? So there's a protein complex which is embedded in the envelope. Why is it important? Like I said, it's a very important, has a very important functionality. So they regulate selective transport of mRNA, ribosomal subunit, and regulatory protein. Right? What did it mean? So because nucleus is the site where your transcription is taking place, and you need these proteins which are synthesized from these transcribed genes, they need to be taken back into the cell. Let me just take you to the uh, this thing here. Yeah, this thing here, right? So this is your nucleus. This is your nuclear envelope, and this is your nuclear pore. This is where your DNA is transcribed, making it a gene, making it a protein, and you need to take it out of this nucleus to take it wherever you want in the cell. That job is very specific because you want only certain kind of proteins to move out, and you want them to go to the destined location. So that has to be very, just like how our cell has plasma membrane, which is selectively permeable. Similarly, nuclear pore complexes also has selective transport, and they transport selectively the the messenger RNA, this ribosomal subunits which translate the mRNA into the protein, and the regulatory protein which makes sure that these proteins are translated. So proteins, so basically the site of protein synthesis is outside the nuclear envelope. The mRNA is made inside the nucleus and is transported out. Okay, right. And then you would be asking, why should be there a regulatory protein? These are proteins which are made outside and then transported back into the nucleus to regulate the whole process. Right. Now this transport of molecule from inside the nucleus to outside the nucleus is through the nuclear pore complex, and it is controlled by nuclear localization signals. As you understand the term, it says nuclear localization. So anything which has a nuclear localization is brought inside nucleus, because the localization, the tag on the protein has a nuclear destination tag, so it is taken to nucleus, right? So you need an NLS. You need proteins which will do the job of pushing it inside the nucleus or outside the nucleus, or in other words, importing and exporting. And since they are doing importing and since they are doing exporting, the proteins are themselves named as importins or exportins, right? And the protein which is absolutely important for this process is the RAN protein. So RAS, RAF, these are all proteins which are very important in cell biology, in intracellular communication, in receptor mediate communication. So similarly here, for the transport across a nuclear pore complex, the most important signaling protein is RAN using a GTP gradient, right? So all of these terms you have already like you heard about. So let's talk about the nuclear localization signal here. So all of this might seem very, like too much information, but these are all just slides that is basically trying to touch upon the topic that you covered in this week. So my intention is to, like cover the topics and then give you some questions so that you understand it better. But anytime if you have doubt, please do put in the chat box. Right. Now, so let me just again move you to the nuclear pore complex before this, because there is something that I did not discuss. So your nuclear pore complex, as I mentioned, I did not mention the structure of it. So let me show the structure here. So the structure of the nuclear pore proteins is basically proteins together are called nucleoporins. You can see the name here, NUPs. Okay. So this NUPs, as the name suggests, they are called nucleoporins. Now, my suggestion to you is, in this kind of specific protein names, the function of the protein is in the name of the protein itself. So just try to understand the protein function. That name will be, it's easy for you to guess. Right. So in nuclear pore complex, the protein is nucleoporins. Nuclear pore, nucleoporins. Okay. Now the structure is very, like very like very categorical. So you have 8-fold symmetrical structure. Okay, so there are 8 times repeating units, and you have 3 major parts. First is cytoplasmic filament, as you can see here, right? Then you have a nuclear basket, which is here. As you can see, it's like forming like a basket. It is all these. So 1, 2, 3, 4, 5, 6, 7. So 1, 2, 3, 4, 5, 6, 7, and these are all coming and connecting together. So this space is here. Okay, so these proteins are covering this spore here. So you have a nuclear basket, you have cytoplasmic filament, and then you have this central transport channel. So let me just show you again. This is a channel, and covering it, you have the cytoplasmic ring, which is formed by the cytoplasmic filament. This is the envelope, and this is traversing the envelope, which is basically, this is your envelope, this bilayer. Then this pore complex is from the top to the bottom. Okay, it's across the channel. Okay, that's important for you to remember. And then you have the basket here, and you have these proteins which are lining the ring. Okay, and this is what makes it the selective. So these proteins that you see here, here, this is a basket, these are the proteins that is lining it up, right? So now let's go to nuclear localization signal and understand what signal and what does it do actually. So your nuclear localization signal is basically a short amino acid sequence. And like I said, localization is signal basically tells the destination of the protein. So NLS directs the protein to nucleus. Okay, and what is the feature of these kind of localization signal? They will be rich in basic residues, so lysine and arginine, and mostly they will be some 4 to 8 amino acid long. Okay, now these are on the proteins. Let me go to this slide. Okay, so let's say you have this NLS and you have this proteins which is with the NLS, and these have to be now moved inside the nucleus. because they are supposed to be taken into the nucleus, right? Okay, so now what is the most important protein for this importance? As I said, the name of the protein suggests the function it does. So the protein name is important. Is basically it is helping in import of proteins from the cytoplasm into the nucleus. Okay, the other name for this is karyoferins. Okay, so now what they do is importance very simply. They have these receptors, so transport receptors that carry the NLS containing protein through NPC. So they basically see, okay, there is a protein with NLS. I need to catch it whole and I need to bring it to NPC. Okay. Now you have two types here. You have alpha and beta. Both of them basically do one thing, uh, making it the transport complex. So you have a cargo. Cargo is basically anything with the NLS. So which is basically here. Okay, so this cargo is recognized by important. It goes and binds to it and it takes it to the NPC. So the protein basically, there is already a tag that takes it to the nucleus. NPC, NLS is a tag, and your important takes it and brings it and pushes it into the nuclear protein complex. Now, the protein that I said is very important, which is here on your slide, RAN GDP and RAN GTP. Okay. So RAN, like I said, all these RAN and RAF and RAS, all of these are very important G protein receptors. So this is also a small G protein means GTP binding, anything which uses GTP currency. The other currency is ATP. So this protein uses GTP as its currency. Okay. So this RAN is a small GTP binding protein. Okay. So when it is RAN GTP, it is in nucleus. When it is RAN GDP, it is in cytoplasm. Okay. So here you can see, if this RAN GDP is there, it is mostly in the cytoplasm. If RAN GTP is there, it is mostly in the nucleus. Okay, that's how it's supposed to be. So now what happens when your nuclear import mechanism is supposed to take place? Okay, this is a very step-by-step process and not a very complicated thing to understand. First thing is cargo has to be recognized. So here is a protein with this nuclear localization signal at its some part of its sequence, and that is bound to the important alpha. First thing to bind is important alpha, and then comes important beta and makes it as this complex, transport complex. Then this complex moves through the NPC, as you can see on the slide here in this image, it moves through the NPC. Okay. Now, once it moves through the NPC, by interacting with the nuclear proteins, it goes inside the nucleus. Inside the nucleus, RAN GTP, which is already inside the nucleus, it binds to the important. Okay. As soon as it binds to the important, there is a conformational change between the entire complex of important alpha, beta, and cargo. And because GTP has now bound to important beta, the beta moves around, cargo is released. Okay, so as soon as your cargo is released, the protein is now inside the nucleus where it's supposed to be. What happens to the importance? It now, because it is now free, it goes back to the cytoplasm, and this is what is happening here. So you have your RAN GTP, you have your this one coming in, your GTP binds to it, it takes it away, analysis is released, and this thing goes back outside. Okay, so that is what happens for importing, and the recycling of the importance back to the cytoplasm is is felicitated by the RAN proteins. So if there is RAN GTP which binds to the important alpha, it gets hydrolyzed to RAN GTP in cytoplasm, and the RAN GTP transports back into the nucleus and reaches. So there is a cycle of RAN GTP and GTP also that keeps taking place to maintain the concentration of RAN GTP inside, because you need that here for this, like cargo release. Okay, so that's why that's how your RAN GTP works. And similarly, your exporting, if you can follow the this thing here, arrows here, exporting is when anything which is to be taken outside. So NLS is nuclear localization signal. NES is nuclear export signal. So a protein which is, let's say it's formed here. Okay, there are some proteins which are here which needs to be taken out. Regulatory proteins, remember? So they need to be taken out. So anything which is has an NES will be bound by the exportin, and then the same way it is taken out. Here again you have a GTP binding. Remember, GTP is all over. It's also there. It is broken down by this complex here to form GTP. So after it moves out, there will be hydrolysis and the exportin and everything will move out. So the same process, import brings it in, export takes it out. It's as simple as that, right? Okay. Yeah, so I think that brings an end to this section because the next section is chromatin remodeling. So let's do some questions so that we understand these topics better, right? So the first question is, which feature distinguishes the eukaryotic nucleus from the prokaryotic nucleus, which I did not touch because I will be discussing in this question. Now, nuclear organization contributes to gene regulation. I told this how, but the nuclear pore complex primary functions to do what, and the nuclear localization signal is what? What is the signal looking like? Please use the chat box to give your answers. I will look at them uh, after a few while. Anyone? Okay. Someone typing. BCCB. Anyone else? Okay, I can see others typing. Okay. People are typing, so I will just wait for some time. BCAB. All right. Anyone must, if you want to change the answer, then I'll just wait for some time. Okay. Okay, BCCB. All right, I'll give you the answer. We'll just move ahead then. Right. So the answer is on the slide. As you can see, the first one says, which feature best distinguishes a eukaryotic nucleus from the prokaryotic nucleoid, which is basically the double membrane nuclear envelope. So now you already know this, that the nuclear material in prokaryotes and is not very structurally compactly arranged, right? It's just a matter which is there in the cell. But in the eukaryotes, you have this very nicely demarcated compartmentalization taking place, where you have the nuclear envelope, which is double membrane in nature, which basically is the defining structural feature of a eukaryotic nucleus, right? And this separates the transcription from the translation machinery, right? Okay. Right. Next one is nuclear organization contributes to gene regulation, as I told you, which is primarily by spatial positioning of the chromatin domains, which is basically, in other terms, euchromatin and heterochromatin. And I also explained to you, euchromatin and heterochromatin basically alters the availability or the access of the gene by the transcription factors, right? Okay. Next one is the nuclear pore complex primarily functions to regulate selective nucleocytoplasmic transport, which is something that we have already established, that there are massive protein assemblies that allow the transport of proteins and RNA between the nucleus and cytoplasm. And the last one here was NLS, or the nuclear localization signal, is basically a short amino acid rich in basic residues like lysine and arginine. Okay. All right. So let's do the next set of questions. Right. First one says important alpha. Remember I told about alpha, beta. So alpha does what? Directionality of the nuclear import is maintained by which of the energy currency? RAN GTP binding to import in the nucleus. So think of the protein, think of the GTP where it is and what happens to it. And carrier are best described as which of these molecules? Anybody has response? Oh, sorry. CBBC. Okay. Anyone else wants to answer? Anyone else? Okay, one more minute and then we discuss the answers. Okay. CBBC. Okay, I'll give the answers and we can discuss. Right. So like you mentioned, most of you, CBBC. So the first question is input in alpha. Alpha's function is to basically recognize and bind the NLS on cargo protein, but this is not yet complete. On this, you need the input in beta to form the entire cargo complex. Next question. You have the directionality of nuclear import is maintained by RAN GDP gradient across the nuclear envelope, right? Remember, GTP is high in the nucleus and GDP is high in the cytoplasm. So because GTP is high in the nucleus, the directionality is maintained. You have that break. So GTP can hydrolyze and, release the important that can only happen inside the nucleus. That's how in the nucleus it has to enter the nucleus for that recycling, right? RAN GTP binding to the importing in the nucleus results in, just like I mentioned, cargo release. So if there is a conformational change in the important beta and the entire complex is then broken up and the NLS containing protein is released. Right, carrier variants, which are also the importance and exportants. These are basically transport receptor mediating nuclear import and export. So these are the imports and importance and exportant. Together they are called the carrier variants. Right. Okay, and the next set of this part. So SNR protein. Now you will have this very very specific proteins names. So the more you try to answer, the more you try to associate with, which is again very simple. So you will get to it, right? So the first one is SNR protein is specifically involved in which of these function? The nuclear lamina, which we discussed, primarily functions to do what? What is the functionality of nuclear lamina? Lamina associated domain, this is what I was telling. This, what will this do? And nuclear compartmentalization enhances cellular function. Why is the compartmentalization at all required? So you have SNR proteins, what would they do? CBBC. Okay. One more minute and then I'll tell the answers. Okay. I'll tell the answers. Right. So these are the answers. Okay. Now the first question is confused on third one. Yes, I will just tell you. Let me just walk you through. Okay. The first one is SNR protein is specifically involved in nuclear import of SNRPs. If you see SNRPs, it's basically how they are pronounced, NERPs. And because the importance is taken in SNR protein, it's called SNR protein. It's as simple as that. Okay. Now, what is this SNRP? What are these analyzed associated SNRPs? These are basically small nuclear ribonucleoproteins. That's why they're called SNRPs. Okay. And what do? They are basically these RNA protein complexes. So we know that RNA is, also has protein as a interaction. So these are those proteins which are involved in the RNA protein complexes. And they basically combine with these pre-mRNA and other proteins. And they're mainly involved in spliceosome, which is basically, if you know, your RNA, which is synthesized, is edited later or spliced to give the final RNA, which is used for your protein synthesis, right? So those kind of functionality is carried out by SNRPs. And these SNRPs, as you can imagine, they have to bind to RNA. So they have to basically be transported into the nucleus, and that is done by these star proteins, nerve proteins. Okay. Next one, nuclear lamina primarily functions to provide structural support and organize the chromatin, which is what I told. These are the lamine proteins. They give the cytoskeletal support for the nuclear shape, and they will then organize the heterochromatin at the periphery. If you remember, the organization of the DNA in the nucleus was in this form, like coiled form. Most of the heterochromatic part will be in the periphery, and the euchromatic parts will be inside the, like, towards the nucleus, right? Okay. Now lamina, which is what the third question is. I hope that is the third question. Yes. Lamina-associated domains are typically associated with enriched heterochromatin and gene-poor regions, right? So LADs are usually gene-poor and transcriptionally repressed. Now, what I told you, wherever they are interacting with lamins already, they are already, like hidden away from the accessibility zones. Just try to imagine like this. You have this DNA and you have these lamina associated domains. So basically domains is part of DNA. When you say a particular protein, for example, lamines here, it basically means that the lamine proteins joins to these regions of the DNA. So that domain, that part of the DNA binds to these kind of proteins. So lamina associated domains is basically those part of the gene which interact with the lamine protein. Now, since they are already interacting with laminin proteins, they basically, I've already covered and they're already extruded, not extruded because that's a very specific term. They're already hidden away from the majority of the section. They're already bound and folded. Now, what you need, these are basically heterochromatin. What you need is basically genes which are exposed so that they can come, the transcription factor, the ribosomes can come and sit onto the genes. Now, these are those regions which are not exposed to or not interacting with any other protein and they are free in their nature. Because your lamin proteins are already bound to these LADs, your DNA now becomes heterochromatin, and these are gene poor. Gene poor means because they are not transcribing, they are not much genes, so they are called as gene poor to differentiate them from the euchromatic region, which are actually the region which gets transcribed. Is that, I hope that is the logic, and I hope that it is clear to you. Okay. And the last question here is, why do you think the nuclear compartmentalization is important? That's because it is spatially separating transcription and translation. And that's why the cell has more control over both of these processes. If your transcription is not correct, then it halts at transcription, doesn't let translation take place. But if it is, there is no checkpoint or there's no regulatory control over it, then if there is an aberrant transcription, it gets translated into the protein and you have a misformed protein, right? So to control such, because cellular machinery or all of these machineries are time taking and very energetically expensive and they have to be very efficient for the proper functioning of the cell. Now, if there is any error in any of the step and it gets propagate further, it is very harmful for the cell. So the cell doesn't want to do that. That's why it wants to be efficient at every step. And what better than compartmentalizing it? So it says that, okay, you make the RNA here. I will check the RNA. If it is good, only then I will send it out. Then it can make its own protein. And there I will have several other checkpoints. So the compartmentalization is primarily helping in these regulatory controls. Okay, right. So let's now move to the next set of organization, which is basically chromatin organization. So we now know that yes, there is like the nuclear envelope is there, and you have your nucleus lamines, and you have this pore complex which takes proteins and in and out of the nucleus. Now let's move inside the nucleus and see what is nucleus mainly consists of DNA. So how is the DNA packaged into the cell? Because DNA is a long biomolecule. A nucleus is a dense and it's a very small part of your cell. So how is this entire big molecule compactly packed into the nucleus is what we are going to do and see how that takes place by chromatin reorganization or chromosome organization. Right. So chromatin, first of all, because that's the chromosome is, if you remember chromosome, it's a duplicated two sister chromatids together, homologous pairs, which basically means homologous chromosomes together. So chromatin is basically the single DNA strand we are talking about, right? So when chromatin organization is being discussed, we are basically referring to how the DNA is packaged, arranged, and regulated inside the nucleus. And like I said, it's a 2-meter DNA which has to be fit inside. The nucleus size is 10 micrometer. So you can imagine the nucleus diameter radius and then imagine how your the 2 meter DNA will fit there. It obviously, if your region is this big, let's say this is your circular region and you have this big protein to fit here, it only has to be coiled first to be even be kind of taking a space inside the hole here right now for that matter. So similarly, for your DNA also to be somehow be packaged inside the nucleus, you need it to be highly coiled. And that's what happens also. And who helps in these? We have the histone proteins, as you can see here, H2A, H2B, H4, H3, H3. So these are the histone proteins. They are basically the proteins that basically bind to the DNA and then help it keep in place, right? Now, why did you do it? As we know, DNA has a negative charge on its backbone. These histones, they are rich in positively charged proteins, right? So there is a positive and negative interaction that takes place between the DNA and the core proteins and the histone proteins. Now, as you can see on this slide here, that so this you can see the DNA is wrapped around these. So you can see if you have multiple of these histones, the DNA, this is a long stretch, gets wound and then again gets wound, so it starts coiling, right? So the core of the complex that forms this binding domain is basically the H2A, H2B, H2A, H2B, H4, H4, H3, H3. So two subunits of each H2A, H2B, H4, H3, right? So these 8 octamer of histone, they basically form the domain where you have the DNA wrapping around. And then you have the linker histone, which is basically here in these strands, which are not part of the spiral, right? So these are basically linked, these are bound by linker histone, which is basically the remaining histone H1. Okay. And these, as you can already imagine, them binding to different regions makes the DNA coiling more effective, right? Okay, so this is how your basically your binding is taking place. So this before I explain the binding to you, let me just tell you what does it form actually when you have this histone and nucleus together, or histone and your nuclear uh, material together. It's called a nucleosome. So remember that if you are talking about chromatin, it is formed of nucleosomes, right? Now, the binding, that's why I said it's a very highly regulated method, because the winding of the DNA is not random. It's a very specific repeating unit. Okay, so when we talk about one nucleosome, which is one repeating unit of a wrapped histone and DNA, we are talking about 147 base pairs. Okay, so these many base pairs, unit of a 147 base pair is part of the nucleosome. Another nucleosome, next 147. Another nucleosome, next 147. So these are involved in the wrapping around of the histone octamers. And if you now just zoom out and try to imagine, you have this one big DNA strand. You can imagine this to be a thread. And you start putting these small knots between each of these parts. So let's say you start here, then you put a knot. So this looks like beads, right? And that's the structure of your DNA. Also, if you look at it under any microscopy, it looks like beads on a string. So your string is your DNA and these beads are the histone that are there, right? And then what is that remaining linker DNA? This one basically takes in around 20 to 80 base pair. So around 220 or 200 base pairs we are talking about for one coiling of DNA, right? So that's very important. And now here is a very important thing that you need to understand, that the turning here, wrapping here is 1.65 times. Okay, so what does it mean? Let's say this is my histone and I start my DNA here. So this is one round, this is two round. That doesn't happen. So 1.5 and some part of it is 0.65 and then it turns away. This is a linker DNA. So next, again, it goes for 20 to 80 and then again it rounds it up for 1.65 times and then again it goes up. So you can have this BD structure on the string. Okay. Now, if the wrapping around histone is very tight, what will happen? It's all imagination. If you imagine, it becomes very easy. If it is wrapping very tightly, it is becoming very small. There is no accessible gene. So this region becomes gene, in this region, you have gene repression. They are not transcribed. But if you have a loose wrapping, you can still have some region which is accessible to transcription factor. That's the region of gene activation. That's where you have the gene regulation taking place, right? So that is a one important concept that you should remember. And that is what gives rise to your euchromatin and heterochromatin, right? So if on the slide, what you see here is, this is your DNA. This is a nucleus, okay? And you start by these long ranges protein. Now see, all these circular or donut shaped, or you can think of them as your woolen yarn, all of these together are again wrapping up on itself. Okay, so you have long range fiber interaction, and then this forms a 30 nanometer fiber. Again, then you have short range fiber. So these fibers are also interacting with each other, and ultimately you have your nucleus packing. So you start from DNA, start binding them up by 147 base pair. Then you have beads on string appearances, and then these histones again start interacting with each other. So the ultimate idea is these beads also start. So if you have this string, you have multiple beads, these beads start interacting with each other, they come closer. Then these two beads start interacting with these two beads. So this gradually it becomes very, very compacted. And that's the whole idea. To fit into the space of the nucleus, it requires to basically have these tight interaction, right? Now, like I keep mentioning about euchromatin and heterochromatin. So let me try to join the dots here. So based on packing, I already told you, if your packing is loose, it's a euchromatin because you have a lot of space for the transcription factor to come. So this is active genes. These are basically gene activated region. Heterochromatin are tightly packed, so they have silent genes and they are at the periphery. So because these are at the periphery, your euchromatin has to happen in the center, right? And then you have, how to identify these regions? You have something called modification, as you can now see on this slide. These modifications you see methylations, acetylation, phosphorylation, and then you also have ubiquitination. These are basically, like the markers on these histones, right? And they tell what the regions are. Okay. So for example, here on your slide, you see that so these are called PTMs. So the histones which are bound to the DNA undergo multiple PTMs, all called post-translational modification, like acetylation, methylation, phosphorylation, all of these. Okay, and these are on the end tail of the histone. Now, what it does, if you have histone acetylation, what will happen? So remember this. Just imagine it like this. Histone is a positively bound, positively charged protein. DNA is negatively charged. Histone positive, negative bind to each other and they have a strong bonding. Now, as strong interaction, not bonding because there is no covalent bond here. Now, what PTM does is, or what happens when the modification happens, you add positive charge, for example, acetylation onto the histone, right? You added more positive charge, which is already present in the histone. Now, as you add more positive charge, it basically neutralizes the positive charges onto. So, when you add acetylation, it basically reduces the positive charges on the histone. Now, because you are reducing the positive charge, the interaction between DNA and histone loosens up, and it is becoming looser, right? So what it means, your packing has taken place and then you have some spots which are marked by acetylation. Because those regions have less positive charge, they move away from DNA and your DNA is loosely bound, so your transcription factors can come and interact. That is why these are called transcriptionally active region. Now, the opposite happens in methylation. Methylation can either increase or decrease, but the mechanism remains the same. If it is to increase the transcription, your DNA has to be available. The chromatin and the histone have to move away, so the binding has to be looser. If it needs to be repressed or not transcribed, then the binding is closer. Okay, that's the idea here. And what are these called? PTMs. They are also called epigenetic, uh, features which are not part of the DNA. So the modifications of epigenetics modification, right? Okay. Now, just a second. Yeah. Another concept before I give you the question is histone code concept. If you remember, this was also discussed in the class. It basically tells, but now histone code, how will the histone code? Histones are basically protein, but they can code because of the modifications that they have. I just told you the modifications, right? Acetylation, methylation. If you have acetylation as the code, you have more transcription. If you have methylation, depending on the context, it can either transcribe or not transcribe. So when we talk about histone code concept, we are talking about specific combination of histone modification. Say you have acetylation and then you have methylation, or you have such kind. This is also a code, right? You have methylation, a lot of acetylation, and suddenly you have a methylation. You have all the possible combinations here, right? So these create regulatory signals, and they determine how gene expression is governed, right? So your cells basically will read these marks and then say, oh, this region has more of acetylation, so I need to remodel this and make it more looser. Oh, this has more methylation, and the region tells that it wants to be repressed, so I need to make it tighter. So histone code basically governs the pattern of gene expression. Will your gene expression takes place or not? Because gene presence is in the DNA. Gene getting expressed or not is based on your transcription factors and your ribosomes. But which gene will get expressed is determined by these modifications which are on the histones, and that is where it's called histone cord concept. Okay, right. And like you can already imagine, why is it very important? Because it regulates gene expression. That is why chromatin organization is an important topic. You understand gene expression, you understand DNA repair, DNA replication because of the packing itself, differentiation and development, because once your gene is packed, when your DNA is packed, it's a cell formed, and then you have your development taking place. But any aberration here leads to cancer, because anything that is perturbing cell biology leads to cancer. And you can have epigenetic problems. Why? Because you have methylation problem, acetylation problem. These are all epigenetic problems, and there are many diseases which are because of epigenetic modifications, right? Okay. Okay, so now I will give you questions so that we can understand this a bit more. Okay, right. The first one is the basic repeating. So I told you already this. If you have to talk about repeating unit of chromatin, what are we referring to? Now, nucleosome core particle, how many histones are there and what else is part of it? DNA is bound around histones in which hand? This I did not mention because I will discuss it in the questions. And histone H1, we know this already. What is its function primarily? Easy questions, not at all difficult to understand. BACB. Okay. Anyone else? BABB. Okay. I'll wait for a minute if anyone wants to attempt. Okay. No one? Oh, someone's typing. Okay, I'll just wait for that person to respond. Can you explain basic chromatin remodeling also? Just basic, not ending advanced. Sure, I will do that. Just let me give the answers to this and then I will tell you chromatin remodeling. Okay, anyone else wants to attempt? Sorry. Yeah, I'm sorry. I think I'm back. Right. So when I say nucleosomes is basically the histone octamers and the DNA wrapped around it, and chromatin is basically all these multiple nucleosomes back to back arranged on the DNA, right? So that's your first answer. Second one is nucleosome core particle. Now that we know that repeating unit of chromatin is nucleosome, what does nucleosome consist of? It consists of a DNA wrapped around a histone octamer, which we have discussed, H2A, H2B, H3, H4, each of them two copies in the octamer. Then DNA is bound around histones in the left-handed superhelix, right? It's a DNA generally is right-handed or left-handed both, depending on which type of DNA it is. In your nucleosomes, it's a left-handed superhelix. Superhelix is tightly coiled and with less distance between the two strands, and it is left-handed. So the mannerism, so this is nucleosome, the mannerism of binding is left-handed. Okay. And then last one is histone H1, which we know is part of the linker DNA. It basically stabilizes higher order chromatin structure, right? Okay. Now, someone asked about chromatin remodeling, which is basically this. When I say remodeling, I'm referred to this reorganization of the nucleosome structures and their positioning, right? Which is basically remodeling is anything which is so in general, also if you're modeling something, you're basically making up the structure. Remodeling is you are in a particular structure and you disrupt it and make it again. So that's what is happening here. And the whole idea of this chromatin, which is basically made up of nucleosome, the remodeling does here is to change the DNA accessibility without changing anything else in the DNA sequence. And if you basically alter the DNA accessibility, you basically change the gene expression, DNA replication because your DNA is loosened up, right? So you need that, and in very simple term, it's basically opening the loosening the structure and tightening the structure with perspective of histones and the DNA. And the whole idea is the looser it is, the more space or the more easily bound it is around the histone, you have more of these RNA polymerase and DNA, which can find the strand of the DNA and come and sit on top of it. Right. If you remember transcription, they require the initiation of transcription takes place because you have to have this region where the ribosome can come and assemble. So that can only happen if your DNA is accessible, and that accessibility is based on the remodeling which is taking place. Okay, and that is marked by the codes that I just mentioned about the histone codes, right? And now when you talk about remodeling, you have several things that is to be taken. They can imagine, right? You need to basically somehow make the uh, interaction between the nucleosomes weaker. So you have to replace the histones with some variants, right? So there is a histone variant SNPA, which I think we should be discussing in the coming slide. Let me check. Yes, right. So what you do is you change the kind of histone protein. With this histone binds to DNA very tightly. You change the histone protein, it doesn't bind anymore. So you change alter the DNA histone binding, or you loosen up the nucleosome by sliding it along the DNA, or you take out the nucleosome and keep that region open like a loop. So you have different kind of way that you can weaken the interaction between the nucleosomes and the chromatin and the histone, right? And of course, these are done by several specific protein families, which I think like if you look into up, you will see the name. But I hope you get the idea that there are several ways on how you can loosen the interaction between the DNA and the histone proteins. Was that clear? You can use the chat box if you have any other doubt. I will, for others, I will just move to the next question set. Right. The next question set is chromatin folding into higher order structure primarily serves to do what? Euchromatin, which is basically the opposite of heterochromatin, is what is it doing? PTM of histone commonly occur on where does the histone modification take place? And when you call acetylation, remember again what does acetylation? Okay, thank you. And when you say acetylation, what does it do? Okay. Anyone else wants to attempt? It's easy question. We have already done this multiple times, so these are not very difficult. BC and BC. Okay. One more minute. If anyone else wants to attempt. Okay. I will give the answers. Right. So, like people have mentioned, BC and BC. So the first one here is basically saying, let me just open it here. Yeah. Chromatin folding into a higher order structure primarily serves to, why do you need this higher order structuring? To facilitate DNA packaging within the nucleus. Makes sense, right? Because it's such a long molecule and it has to be packed into micrometers of cell nucleus. So that's why you need this dense packaging. Okay. Next one says euchromatin is typically characterized by conformation, open conformation and active transcription. This you need to make a mnemonic, or you need to somehow remember what is euchromatin and what is heterochromatin. Associate euchromatin with active transcription. One terminology, like say active transcription or gene rich regions, should enable you to remember the other one as well, right? Okay. PTM or post-translational modification of histones occur in histone tails, as I mentioned while discussing, right? So all these phosphorylation, acetylation, methylation, ubiquitination, whatever you name it, happens on the histone, that too at the N-terminal of the N-terminal tails. N-terminal tails, okay? And the last one is histone acetylation is generally associated with increased transcriptional activity. Why? Because acetylation, which is basically adding of the acetate group, neutralizes a positive charge. And because the positive charge is reduced on the histone protein, they loosen up the interaction with the DNA, which is again a negative charged molecule. And because they loosen up interaction, you have more of the transcription machinery taking place. Okay, the last set for this. Okay, the histone code hypothesis, what does it propose? SEND-PA, that's what I was telling. And what is it described as and what does it do? And where is it present? And which of the following describes the nucleosome modification? Like, what is the role of it? Okay, for C. Any other answers? Any other responses? Triple BC. Okay. 1 minute. These questions are not, like they're not. I hope they're not very difficult. If so, maybe what. So I always tell that you can try attempting at your end. You necessarily don't have to write in the chat box. But when I am discussing the answers, I hope you can trace back and think that where you're thinking in the correct directions. So that's why even if you think you don't know the answer, try to think on the answer and think of the possible reasons of what option it could be. Okay, let me give the answers and then we can move. So B, 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 and C, like someone mentioned, right? Okay, so the first question here was the histone code hypothesis. Remember, we were talking about the acetylation, methylation, and all of these on the histones in the nucleosome. So they basically are a specific combination of histone modification and they regulate gene expression. If you have multiple acetylation back to back, so you know that region is transcriptionally more active. If you have acetylation less but methylation more, you might have repression of that expression there. So they basically govern the pattern of gene expression, right? Then you have the CEN-PA, which is best described as a centromere specific variant of histone H3, and I will tell you what does it do. And then you have the next question, which is presence of CEN-PA at the centromere is critical for chromosome segregation during mitosis. This we had already discussed in the previous problem solving session. Why is CEN-PA important, right? And the next one is, which of the following best describes the role of nucleosome modification, dynamic regulation of chromatin accessibility, which I think has been now fairly established, right? Now let's just to complete the CEN-PA questions. So CEN-PA, as the name suggests, is a centromere protein A. Okay. Now this specific protein is only present in the centromere region of the DNA. So you have DNA. Centromere is the the central region of your chromatin. Okay, so you have your uh, chromatid, center is just centromere, and you have this protein which binds to the centromere. Now, this one, CEN-PA, it binds to H3 variant. Remember, I told you that there is H3, but if you have a variant of H3 in your nucleosome, you have the CEN-PA coming and binding it. Okay, so the histone H3 near the centromere region, it binds to the NPA. Okay, and what it does, it basically uh, uh, how do I say? It gives an identity to the centromere, saying that hey, I am here. So now uh, this is CNPA is basically marking that region. And what is it marking for? It is basically marking it for making it a kinetochore. Okay, and kinetochore, if you remember, is basically involved in the segregation of the chromatids during NFAs, right? So your CEN-PA containing nucleosome basically recruit other kinetochore protein, and they have this spindle mitotubules. And once that happens, you have these spindles connecting to the kinetochore, and they pull them to the opposite poles, right? So you the centro PA is basically important for segregation. Now, if there is no CNPA, you don't have a kinetochore and you don't have chromosome segregation because there is nothing that pulls. The microtubule doesn't get a region to connect to and pull the chromatid towards itself, right? That's how the chromatids are pulled. So what happens is you have genomic instability and it gives rise to your genomic problem, for example, Down syndromes or any such problem which associated with unequal distribution of chromosomes in your daughter cells. So CEN-PA is therefore very important and it is like it is not only relevant for the functionality of the cell, but it's also important from a medical perspective. So that's why CEN-PA, as it is written here, it is important for chromosome segregation at specific variant of histone H3. Okay. Right. Now let's move to the third topic of this week, which is SMC protein, which in many different ways we have been discussing for the past many sessions also, but this time in a bit more descriptive way. Okay. So this was the like the longest topic in this week's lecture notes. So I hope you bear with me, and then we do enough questions so that we can, like cover up the syllabus. Okay. All right, so let's begin. So SMC protein, as you know, is structural maintenance of chromosomes. The full form of SMC is structural maintenance of chromosomes. So anything that is maintaining the chromatin structure and chromosome structure, right? And these are ATP-dependent molecular machines, right? For keeping the chromosomes in their correct place. So you basically not just require them for keeping them in the correct place, you require them for sister chromatic cohesion. Remember, sister chromatids are basically the DNA replicated part. You have these two chromosomes, they are maintained, they're kept together by SMC proteins. If you will read those names, if you remember condensin, cohesin, all of these are SMC proteins, right? So you have these sister chromatic cohesion, you have chromosome condensation, which is taken by condensin. Why do we need condensation? Because when we are doing cell cycle, the packing of the cell has to be dense. How the that's how condensin functions. They help in packing of the cells, DNA, sorry. And then you have DNA repair, and which was the ultimate functionality, structural maintenance. So you have DNA repair and genome stability, right? Now, if you look at these proteins, this is your condensin, this is your cohesin, and the other name or the other. So basically, there are three type of SMC proteins broadly: condensin, cohesin, and it's also called, the next one is called SMC 5 and 6 complex. Okay, so on your slide right now you have condensin, cohesin, and this SCC, SMC. These are very complicated names. Sorry, this is a very complicated name. It looks like very complicated name, but what you should try and remember is the functionality and specific uh, uh, places where they are functioning. Okay, sorry, anything happened here? Yeah. Right. So now, if you see any of the SMC protein, they will have one ATPase head domain. Remember, they use up ATP. So for their any of this structural maintenance, so they will have an ATPase domain because that's where they will hydrolyze the ATP. They will have these long coil arms. Coil arms means these supercoilings. You can see multiple coils. So coiled coil, not only one coiling. One coiling is called coiled. When you have the coiled form further complicating and turning on itself is called coiled coil, supercoiled or coiled coil, right? So you have a coil long arms, and you have this hinge domain, which is here. Okay, this hinge domain is also there. Okay, all of these you will see this structurally very similar kind of protein, sorry, structure, that they will have hinge domain, which is basically the V shape. So you have this V shape, you have supercoiling, and you have this ATPase head domain. Okay, now condensin, cohesin, even though they have a proper name, they are also form of these 1, 2, 3 complexes. So cohesin has 1, 3, condensin is 2 and 4, and the remaining 5 and 6 goes to its unnamed complex. Right? Cohesin, we know the functionality, so we gave it a name. Condensin, we know the functionality, so we gave it a name. But 5 and 6 have not been explored to that content great height, so they are just yet unnamed, but they function in DNA repair and genomic stability. Okay. Now, just for your revision, once again, the cohesin complex has 1 and 3, as you can see here. Yeah, it looks too much, but just bear with me. So cohesin has 1 and 3, and you have these other proteins, SCC and SCC13, which are basically giving the structure. What you need to remember is the function of these proteins. They hold the sister chromatid together after replication. Why do you need that? Because they will be the one that will split in mitosis. They will go in the opposite poles, right? But before that, you need to keep them in place to line them up in the metaphase in the equatorial plate, right? And your cohesin keep them intact till they are separated, and they are basically important for genome organization, metaphase, basically. Okay, so this was your cohesin. Now coming to condensin, which is here, you have 2 and 4, and then you have these regions. So these are basically accessory proteins which make up the function of condensin. Condensin, the name suggests, what does it do? It condenses the chromosome during mitosis, and then it does something which we will do in detail in the next few later slides, which is called loop extrusion. Okay, don't forget that. We'll do that. And then it basically ultimate the most important function is it helps the proper packaging of chromosome during cell cycle. And then the last one, that is SMC 5 and 6, that as the name suggests has 5 and 6 as the core subunit. And then you have these NSE 1 to 6 as the additional subunits, which is basically important for sumo ligase, right? Sumolation. I will explain these when we do the question. I don't want to burden you with much more information, but these are more important for DNA repair, right? So there are three broad classes, and each of the classes have their own functionality. And what you need to remember is a component in each of this and what are the functionality. So as we do more questions, it should become clearer. Okay, so let's do that so that we catch up on the questions. First one is SMC proteins are primarily involved in which? This is a basic question. Next, structurally SMC protein can be characterized by which of these structures, right? The cohesin complex is primarily functioning for what? And when you say the core SMC protein of cohesin, so these are the kind of question that you will get anyway. Core proteins of cohesin, core protein of condensin, SMC56. So these you need to remember. Easy questions. It's okay if you can't, if you're not able to answer right away, but just try to, like read the question and see what kind of questions will come from this part of the syllabus. Triple BC, anyone else? Okay, let me give the answers. So the first question is SMC proteins are primarily involved, as we have already discussed, that they are important for chromosome organization and stability, right? So next one is structurally SMC proteins are characterized by this. We have already discussed. This is coil arms with ATPase head domain. So you have ATPase head domain, the arms, and you have the other hinge regions, right? Okay. The cohesin complex functions in sister chromatid cohesion. That's the cohesing. There is a cohesive activity. Okay. And cohesins, SMC subunits core are the 1 and 3. Okay. This was easy. Next one. Sorry. Non-SMC subunits of cohesin. Now, every SMC protein has a core SMC protein and then non-SMC subunits. So the non-SMC subunits also you need to remember. So for this one, what is the subunit, non-SMC subunit of cohesin? Cohesin establishes cohesion during which, this is again very repeated question. We know this already. Cohesin complex can be sumoylated by, and sumoylation is what? What I told you that I will discuss about the sumoylation. So I'll do that here. Anyone? Okay, I see someone typing. Okay, 2B and 4B. All right. Anyone else wants to try? One A. Okay. Anyone else? Okay. No worries. I will give this answer. Just remember to read them up at your end for the exams. Okay. Non-SMC subunit of cohesin basically involves the MCD1 and SCC1 and SCC3. Okay. So yeah, you might not want to remember the full form, but what you want to do is you for sure want to remember the combination. So 1, 3, SMC, SCC1, SCC3, these you should try to remember because that's what will come handy in these exams, and that's what will mostly come in your. So most of these questions are from assignments, last year assignment exams. So try to remember the combination. That's more important. I can understand why there are no responses or why probably you have a lag, but that's all right. The whole idea here is that I've given this whole repository here. Whenever you're revising for your exams, you can come back to these videos and PPTs and revise. Right, they're all in one places, right? Okay, the next one is cohesin establishes cohesion because you need to do cohesion. S phase of the cell cycle because that is the DNA replication. That is when your second sister chromatin comes out, and when your sister chromatin comes, you have to be bound to each other. So you need cohesion, right? Okay, the cohesin complex can be sumoylated by here. The answer is MMS21 or it's also called NSE2. Okay, so basically this enzyme is a sumo ligase and it modifies the cohesin, right? And what is this sumo? Sumo basically means small ubiquitin-like modifier. Okay, so we already know what is ubiquitination doing, right? So when you have cohesin sumolation, is basically you have the sumo proteins attached to cohesive subunits, and then it basically fine tunes the activity of the cohesin, right? So it is involved in the regulation of gene expression. So it basically marks it for uh, degradation, or it removes it from the binding region. So basically, uh, these is done to complex of cohesin by this particular protein, and you need that to be removed also, right? And it influences, as I just mentioned, because it will influence how they are binding to the proteins, how they are binding to the DNA. That's why it affects the stability and the function, right? Okay. Next set of questions. You can just now look at the questions and try to make a mental note of it. But like you know, this PPT will be available to you as both PPT and as recording of the lecture. So you can always revise later, right? The first question here is the cohesin complex can be sumolated. So, I'm sorry. The condensin complex is mainly required for what? So now we are now moving to condensin. We have done with the first set of SMC proteins, which is cohesin. Now we come to condensin. The first question is what is it required for? Second, what is the core component, SMC and non-SMC? The same kind of question. And then what is the activity mostly important for? You can see that, like I said, it's a pattern. If you remember for one, you remember it for the other as well. BCAC, okay. All right. Let me give the answers. Right. Like people have mentioned BC and AC. So the condensing complex is basically required for condensing, which is basically chromosome condensation during mitosis. Right? Okay. The core SMC protein that you need to remember for condensing is basically 2 and 4. 1 and 3 cohesin. 1 and sorry, 2 and 4 is for condensing. Okay. And next one, the other non-SMC subunits here are the cap, H cap, we saw in the slide also, right? So the cap proteins, these are basically again for the chromosome compaction and activity. Okay, and the last question was condensing activity is especially critical during mitosis and chromosome segregation because it's about chromosomes condensation, right? It's easy question. Let's do the next set of questions, which is for the next set of SMC proteins, which are 5 and 6 complex. So what are their function? I already told you once. What are their core subunit? What is the additional subunit onto the 5 and 6? And now this name, what does it do here? So same activity, we have already discussed this, so it shouldn't be very difficult. BCCB. Okay, anyone else? Okay, BCCB, right? Let me give the answers. Okay. So, like I already mentioned, the SMC 5 and 6 complex is for the activity of DNA repair and genome stability. The core SMC proteins are in the name itself, 5 and 6, right? And the additional subunit here are the NSE proteins. NSE 1 and 6. 1 through 6 means 1, 2, 3, 4, 5, 6. Okay. NSE, just for your information, these are the non-SMC elements, the same kind of question that we have been doing so far. And now the NNC2, which is MMS21, in the complex will do the same activity as it was doing for the cohesin. It is basically a sumo ligase. So it will act as this ligase, which will add the group of ubiquitin onto these SMC components, and which ultimately act, they regulate the DNA repair mechanism, right? The same kind of functionality. If you remember one, it becomes easier. Okay. And now we go to this very interesting topic, which is basically DNA loop extrusion. And we see why is it important. And this was a short lecture in this week's lecture notes. So we will just try to understand what is DNA loop extrusion and what does it do. Okay. So, right, let me just put you on this slide here. So, right, this is basically DNA loop extrusion, and let me explain to you, try to explain to you what we mean by DNA loop extrusion, right? So, DNA loop extrusion is basically a mechanism by which the SMC proteins, the ones that we have just read about, cohesin and condensin, how they organize the chromosome. We have been talking so far about these two SMC complexes, uh, responsible for organizing the chromosomes, genomic stability. So we'll see how they do that, right? Okay, so the first thing here is what does the word exactly mean? Okay, so it basically means that it is so it's an ATP driven process. First of all, the energy required process where you have these complexes, they bind to the DNA and then they progressively reel in DNA to form a growing loop. So you can see it like this. Okay, so you have these proteins, nucleosome, RNA polymerase, all these proteins here. And then you have the structural loop extruders such as condensins are placed here. And then they keep reeling in. You don't know what is reeling in? Reeling is basically like those reels of kites that you have, right? You put and then you start pulling it towards yourself. That's exactly what is happening here. You have these proteins here and then they keep winding in and bringing this DNA strand closer to it its own. So now you can see here, let's try to understand here. You have this SMC loop extruder, so-called here, and it is now reeling in. Okay, so what it is doing is bringing this nucleosome closer to each other. What is happening in the process? This loop is getting bigger. So this is getting extruded. Extrusion is basically putting it outside the surface, right? So in the process of reeling in the DNA towards itself, it is pushing these part of the DNA out of the loop, right? And that is what it ultimately does. When it brings it along itself, it can also bring it along. It does it for the stability of the structure. So that's this whole coordinated step. So the extrusion basically means that it is pulling the DNA out and making it as a loop. Okay. So not by passive folding, but by organizing the molecular motor. So it pulls and basically it's like a reel. So a reel which is moving towards a particular protein and then moving out, right? Now we already have seen that SMC has two SMC proteins forming a hinge, has ATPase head domains, has coil arms, right? So now what does it do? Let's understand the step-by-step process of a loop extrusion. The first step here is loading. Now what is loading? Loading means you have a condensin or any other cohesin or any of these SMC proteins, which is first coming and sitting onto the DNA, as you can see on the slide already, right? So it is coming and sitting here, or here, wherever you want to see it, right? Next step is DNA entrapment. So there is this ring. Remember, we already have seen this multiple times. Let me go back to this step here. So photo here. It sits and it forms a ring. It sits, it forms a ring. It sits and it forms a ring. So the first thing it will do is it will sit. And then the next thing it will say is, hey, I have this ring. So let's start putting the DNA through this ring. Okay. So the next step is DNA entrapment. Third is ATP binding. So it needs to use up energy. For that. It will bind ATP in its head domain. And as soon as it binds to the ATP, there will be a conformational change in the whole complex. Okay. Then what happens is you have DNA translocation. Now, as soon as your conformation moves, your DNA is now pulled towards the ring further and it start becoming a loop because it has to pass through this and it start becoming a loop, right? Now, it can pull it both side, like both side meaning this here and here both, or it can just pull from one side. You can imagine it. For example, you have a hole in your garment and there is this neat thread coming out and you pull that thread out from that particular nozzle. So when you take it out, it is coming out like a loop. So that's what it's happening here as well, right? And then once it has started forming a loop, it will continue to enlarge the loop at a rate of around 100 to 1000 base pair per second. So we'll keep pulling those many bases out. So when does it stop? It stops when it encounters a signal. What is that? Uh, Presence of another SMC complex. So there is one SMC here. Let me show you in the slide. One SMC here, one SMC here. It keeps pushing it out. It's extruding until it reaches another SMC complex, which is on nearby, or it reaches a so if you have read transcription and translation, you know there are regions in the DNA sequence which are specific for region for stopping the particular process, be it transcription, translation, or any DNA replication. Similarly, here you have these protein boundaries. For example, you can see here CTCF. If this encounters CTCF, then it basically stops the the growing of the loop. So the process halts there, right? And because it is using ATP, it basically, if it reaches another ending point, it stops utilizing the ATP altogether, right? So I hope you have understood the process of loop extrusion. Now the only question is why to do this at all. So this is done because it is helping your organize the genome into domains. What domains are we talking about? We are talking about these domains here, right? This arrangement of the DNA as such that it is looped out, is more available. So it is basically responsible for um, organizing into particular domains. It also is important for condensation, as you can imagine, it is folding upon itself and bringing it closer to each other. And it is important for genomic stability because if you loop it out, you stop the entanglement of the DNA strands. You can replicate, you can coordinate the replication. So you can, there are certain regions which will be replicated, the others are under tight tension. So if you have more, this is also very easy to understand. If you leave a strand just like that of wool, it will become very coiled structure. But if you put it in a loop structure, it is not coiled and it is stable. That's exactly the thing in DNA as well, right? So basically, condensin and cohesin both do the same activity of DNA extrusion, but albeit difference in their how, what is the speed, when do they do it, and what is the boundary control, right? For example, cohesin, it basically has a very slower extrusion. It posts the DNA very slowly, and it does it in the interphase stage. This is just before the, like just after the DNA replication, or probably in between the cell cycle. While condensin does it explicitly in the mitosis step, right? So these kind of differences are there. And if this, you can imagine, if this is not done properly, it can lead to several genomic instabilities and cancers and missegregation of chromosome, right? So it's all for the maintenance of the chromosomes, right? Okay, so we'll do questions so that you get more hold of this topic, and this will be the last set of questions, like in this next few slides. Right. So let's do this quickly. DNA loop extrusion by SMC complexes primarily contribute to what? So I already mentioned this. Condensin mediate loop extrusion requires which of these activity? The rate of DNA loop extrusion by condensin is which one of this? And during loop extrusion, what does condensin act as? These are not something that I discussed, but then these are not something that are very difficult to understand. So maybe if you just think through the question, this should be easily attemptable. Hmm. Anyone? BAAB, okay. All right. Okay. I'll give the answers. You can check at your end. Okay. It's B, A, C, and B. Okay. First one is DNA loop extrusion by SMC complex contributes to chromosome organization into loop. This was easy to understand because that's what we have just mentioned, that they basically loop it out and they organize the genome into these structured domains, right? Next one, condensin mediated loop extrusion. It requires ATP hydrolysis because all of these SMC proteins have this ATPase arm, right? ATPase head rather. Okay, now this one, the rate of DNA loop extrusion, this I did not mention, but I mentioned in passing. This is hundreds to thousand of base pair per second, right? So condensin does it at this rate. And during loop extrusion, condensin acts as a molecular motor translocating along DNA, right? It is not a binding protein. It is definitely not a polymerase, and it's not a histone chaperone. It is working on moving the DNA along its way, right? So that's why it is the correct answer here is it's a molecular motor. Okay. Okay. The next set of questions. Single molecule studies. Okay. Now remember these terms have come from the lecture notes. So single molecular studies suggest that the number of active condensin complex per chromosome is okay. An anchor point in loop extrusion refers to, so the technical terms which I did not use because I knew they will come up with the questions. Loop extrusion helps in mitosis how, and termination of loop extrusion can happen when we are talking about condensin. No one? Okay, let me give the answers so that we move quickly ahead. Okay, so here the first one is single molecule studies, which is basically how you study how you get to the nitty-gritty of these methods is basically when you take a single molecule and you study them in detail, right? So single molecule studies suggest that the number of active condensin complex per chromosome is limited and dynamically regulated, right? So basically, in this, they are present at certain numbers, and that they're dynamically loaded and unloaded depending on which cell cycle stage, because they are limited in number and you can only use certain at certain stages you use certain kind of condensin, so you don't load everything or you don't unload everything, so you basically regulate the number. Okay, now anchor point in a loop extrusion, it refers to a fixed site where SMC complex remain attached while extruding DNA. It's very simple. It is the point where your SMC will go and bind, because for any region where the DNA has to extrude out, it needs the SMC has to first sit. So the place where SMC goes and binds, or SMC helps itself fixed, is the anchor point. Okay, so it's basically the region where SMC is held together, right? And the loop extrusion helps in mitosis by compacting chromosome for segregation, that we've already understood. And the last one is termination of loop extrusion can occur when condensin encounters another complex. This is what I mentioned, either another SMC or CTCF. Okay, any motive for that matter. Okay, now as you can see, the question type changes. This is because this is from a previous year's assignment. So let's try to do this quickly. So pick out the correct statement regarding the role of condensin complex in chromosomal structural maintenance. Anyone? Okay. So the answer here is C and D. It's basically talking about condensin, right? So the first one is talking about tethering DNA region between sister chromatids, that is cohesion. So cohesin. So that is for not condensin. The third one is for condensin, the complex tethers DNA region within chromatids because you're condensing them, right? So in the chromatid, you are condensing the complex. So C is correct. And since here, you don't have to worry about the second option also because you know that A is not correct, C is correct. So this was the correct answer. Yes. So since you are, that's what I'm telling. Since you are sure about the third statement and you know this is the only option that is correct, just go with it. Okay. Next one. How many condensing molecules act on the loop X? This is very easy. How many do you need? How many molecules should come for extruding the DNA? And which is the wrong statement here about SMC protein complexes? Okay. Anyone? Let me give the answer here. B and C. Sorry, B and B. Because if you remember the extrusion, you need just one molecule. The moment it encounters the other molecule, it stops extrusion. So one molecule is enough for extrusion. And second one, everything is correct except for this because SMC protein complexes are found in every domain on the planet. So you have it in bacteria, archaea, eukaryotes, everywhere. So it is conserved across all life forms. 1 and 2, which is correct for 2, yeah, I mean, one, I hope this was the one that you were referring to. So only one molecule for extrusion, right? Okay, next set of questions, which are basically, yeah, 3 more slides and then we are done. So so these are repeating questions. It might seem like a repeating question, but that's because these are taken from different assignments. The role of cohesin during DNA repair, which histone protein is not a core component of the nucleosome. Which of the following correctly describes heterochromatin and which histone variant is specifically associated with centromere? This you already know. Okay, ADBC. Right. I'll give the answer so that we can read quickly. And it was correct. Role of cohesin. Again, cohesin means two sister chromatids or chromosomes. So it facilitates fidelity of homologous recombination by holding sister chromatids together. Remember, meiosis, sister chromatids are held together. It is only at the sister chromatids, one pair from each of the homologous chromosome which undergoes the recombination, right? Okay. Histone protein is not a core H1 because H1 is part of your linker DNA. Which of the following describes heterochromatin? Euchromatin is your active, right? So heterochromatin is basically your transcriptionally inactive. So silent, and that's because it's tightly packed. And this we discussed in length that the centromere histone variant is CNPA. Okay, second last slide. Which technique is used for real-time imaging of DNA? So basically in the guest lecture, what did the professor mention about the technique for real-time imaging of DNA loop extraction? How do you actually see it? Which process powers the loop? This we already know. What may happen when SMC proteins fail to properly function in cell? This is something I've been telling you. And what does the role protein WAPL play in cohesion dynamics? Something that was covered in the lecture, guest lecture notes. Okay. Let me give the answers. So the first one, I think you remember from the lecture, no, that the method that they had used was single molecule. Remember single molecule we discussed already, and it was a microscopy because I told you in microscopy only we can see the structures. Which process powers the loop extrusion? ATP hydrolysis, right? Okay. Now, what may happen when SMC protein fail to properly function? I told you if the SMC proteins are responsible for chromosome organization. If that doesn't happen, it is missegregated and there is genomic instability. Just remember the word genomic instability. See the other option, it says it condenses more efficiently. Of course, not correct. Replication can't speed up if things are not working fine, and cell can enter apoptosis immediately. That is not correct because there is nothing to guide it for apoptosis, right? There is no repair mechanism working there. And the last one is WAPL. It's basically unloading cohesin from DNA, which is basically when your cohesin is on the DNA, how to remove it and move to the next step is when your WPL protein. Because remember, if they're loaded, they also have to be unloaded. And those, when their extrusion is done, how do they take it off? So they have recognized, okay, there is an ending point. Now, how do they move ahead, away from the DNA? That's when these kind of proteins come into action. Okay. And the last slide for today. Which protein works with cohesin to define TAD boundaries? First of all, we'll just do that and then I'll tell you what is it. An SMC protein forms which structural configuration? Anyone else? The last question. Okay. So DNB. Remember CTCF I already discussed. TAD boundaries. What is TAD? Topologically associated domains. We also did lamine associated domains. Okay. So it basically, in other words, it just meant that how does cohesin know when to finish? It when to stop the extrusion. Okay. And that works with CTCF. And SMC proteins, as I have told you, they form this coil structure. So with this, you should have guessed it, and I think you did it right. Here, we already had discussed CTCF, only that we did not know TAD. TAD is topologically associated domains, right? Okay, so that brings an end to this session. If you have any doubts, yes, you can please type in the chat box or you can ask also. We might not have a session on next Monday, which is 9th March, because of some problem that is upcoming, probably. So if the class is rescheduled, you will definitely get to know with the mail. Right. So most probably this will be held in the Fridays or Saturday. You will get the mail accordingly. If not, if you don't get a mail, the class takes happens next on next Monday, same time, 6 to 8. right? Uh, yes, lecture material in the sense, do you want the PPT? Are you talking about the PPT? PPT, yes, you will definitely get PPTs, and these recordings are provided to you in a link, so you will definitely get that.